still hear me, of course, despite the probably the heavy rain. So hopefully you can still hear me, and of course you can later on see my presentation on the screen. So let me now start immediately before we encounter any problem due to the heavy rains. Okay. So we're now on session 3 for this week. So let me, all right. So there, so let's now start. So for this afternoon, we will be focusing on, okay, we will focus on non-Mendelian genetics. Now, this is actually the a pattern of inheritance that is dedicated for grade 9 students. So what we did for the previous 2 days are all revisits of the things you've discussed and learned from grade 8. So how does it relate to what we will learn this afternoon? Later on, we will understand it better. So, for this afternoon, we have two objectives, and that is first, we need to know and be able to differentiate the different patterns of non Mendelian inheritance or non Mendelian genetics, and of course, understand and be able to explain how they are different and how they are similar to one another as a pattern of inheritance. So let's recap regarding important concepts regarding or that is our foundation for our discussion. So please take note when you talk about trait, it is the category of a certain observable characteristic. And when you talk about an allele, it's actually the option or the variations for that particular trait. A very good example for this is, of course, when you talk about seed color, you are actually referring to a trait. And then when you talk about the available options for a particular seed color, either yellow or green, you're actually referring to the alleles or option on that particular trait. Now, we also discussed yesterday about the difference between phenotype and genotype. So when you talk about or when you're asked for a phenotype, you're actually asked to simply give the observable description or give the observable characteristic or give the description. And of course, when you are giving a description, usually you use descriptive words or adjectives. So when you talk about genotype, it talks about the genetic material. And of course, it's very difficult to represent the genetic material. So the best way to do that is to simply express or represent it using specific letters that are actually used as code for the genetic material or a set of genes. Another thing that we also need to remember when we use the terms homozygous and heterozygous. So homo, the prefix homo states or refers to a state of being similar or same. In this case, we have same letter capitalization or same letter cases. So it's either when you talk about homozygous in terms of its application to the genotype, it's either capital A, or a small letter A. So same letters, but and for both letters, you have the same capitalization or cases for the letter. However, when you talk about heterozygous, it refers to the state of being different or opposing or contrasting to one another. So in this case, you only have one possible representation for a heterozygous. It's a combination of the capital letter A and small letter a. All right, so hopefully that's clear to everyone. We already recap trait and allele, phenotype and genotype, and then we have also homozygous and the word heterozygous. So another important thing that you need to remember is that when you talk about Mendelian genetics, this is also commonly known as complete dominance. Okay, let me explain why is this called complete dominance. Now, of course, Mendelian genetic gives recognition to the individual or to the person of Gregor Mendel, who was able to initially propose the concept of dominance. However, based on his work, based on his study on the pea plant, he was only able to assume or give the summary or give a conclusion that for every trait, there are two alleles or options. One is dominant and the other one is recessive. However, this dominant allele has a complete ability to hide and mask the presence of the recessive allele in a heterogeneous genotype. So meaning, let me give you an analogy. Let us say this open palm here, my left hand, is the dominant allele. Okay, it's representing a dominant allele, a capital letter. The other one, my closed fist, will represent the recessive allele. Okay, now let us combine it together. Let us say we have a heterogeneous genotype. Can you see my closed fist behind my left hand? Yes or no? No. So in that manner, or in that principle, or in that scenario, this dominant allele, which is being represented by my open palm, has the ability to hide the presence of the recessive allele. The recessive allele is there, present, however, it is not shown. It is not expressed on the organism because it is being completely hidden. Okay, but it doesn't mean that the recessive allele is not there. It's just that it's not expressed, it's not shown on the organism. Okay, so that's when you talk about complete dominance. Now, why is it significant, teacher? Why we have that to with why need to emphasize that? We'll understand it later on. So yesterday we focus on three problem set. The first problem set talks about the seed shape. And of course, you actually had this result. If ever you were not able to screen capture the presentation yesterday, it's here. Okay, I'll give a few seconds. So that's for problem set number 1. Okay, how about problem set number 2? It talks about the length of the hair of a certain breed of cats. So, and this is our solution for problem set number 2. By the way, it's problem set number 2. I was not able to edit the label or the header for the slide. Okay, for those again, same thing. If for those who are not able to screen capture, I'm giving you a few minutes or few seconds rather to take a picture if ever you need a copy. All right. So for problem set number 3, this time it talks about the shape of the earlobe. So it's either free hanging or we call it detached, and the other one is attached earlobe. So same thing, I'm showing you again the solution for problem set number 3. All right. So hopefully you now have your own copy for the individual problem set. By the way, the problem set, all of the questions are found on our module. So you can always go back to that. So let's now go back. Let's now focus our attention on non-Mendelian genetics. So the word non there sometimes give us a connotation of being negative or the word not Mendelian. Okay. So let's clarify that. So in our previous discussion, as I've mentioned a while ago, we emphasized the importance and the interaction, how dominant alleles and recessive alleles um if has a relationship, and at the same time how they one allele affects the other. So as you can see here, we have a homozygous dominant What is expressed on the organism is, of course, the dominant option. If you have a heterozygous genotype, just like this one, and this here, yes, it has a heterozygous, meaning a carrier of the recessive, but what we see on the seed shape is actually, or what is expressed on the seed is that it's round, just like your homozygous dominant. But they differ in terms of their genotype, meaning their genetic material. And then you can only see the dented or wrinkled option for this trait or characteristic only if you have both letters in small cases or lower cases, representing that it's a homozygous recessive. Because again, your dominant allele has a complete dominance over recessive, meaning na siya complete superiority over the other option. However, that is not actually the case. Mendel's work, it was just at the starting point or the tip of the iceberg in discovering other patterns of inheritance. Complete dominance is just one out of the many patterns of inheritance that was later on discovered by other scientists with the help, of course, of the modern technology. So, here we have an example of other patterns of inheritance. So, for this afternoon, we'll focus on the other and new complex patterns that are collectively called as non-Mendelian inheritance. So take note, when you talk about non-Mendelian inheritance, it doesn't mean, or genetics, it doesn't mean it's against Mendel. The concept here is that this group of scientists or this group of pattern of inheritance that we will talk later on are actually um, concepts that were developed after Mendel. So if you look back on the life of Mendel and his work was not immediately recognized in the scientific community. It was actually later pa after his death that the the scientific community later on had interest and find his study, his conclusion very relevant. And of course, because during his time, modern science was not really that um high tech. But now, with his study and together with the high tech tools and technology, scientists were then further improve, was able to further improve his concepts of genetic or genetics. Okay, so what does that mean? There are actually two major, again, as I've said, there are other patterns of inheritance, but for in our case, we'll focus only on two non-Mendelian genetics. Again, the word non-Mendelian means the improved version of Mendel's work. But it doesn't mean that they do not agree with Mendel. It's just that they were able to find and expand his observation that it's not just limited to complete dominance. And one of that is what we call incomplete dominance. So I have here an image to help you visualize what we mean by incomplete dominance. Let us say if this is again my right hand, the dominant allele, let us say red, and my closed fist is my white option. If I follow complete dominance, what color do you see? Red or white? Again, if we follow complete dominance, if my red hand is my left hand is red and my closed fist is white, what do you see? Okay,
Red.
red. Red. So that's complete dominance. That's what you already know from grade 8. Now, aside from complete dominance, this is another concept, we have incomplete dominance. So let me give you the scenario here. Again, going back, this is the red. Okay? The red option. Again, this is white. However, this time, what do you notice on my dominant allele?
May openings too.
Okay, very good. As you can see, the space in between my finger is now obvious. So because of that, what happens to the ability to cover? Is it complete? Is it full?
No,
No,
sir.
very good. So that means my dominant allele does not have now the complete ability to cover my recessive. So what happens now? Can you see the recessive behind my dominant allele? Yes or no?
Yes.
Yes, you can now partially see it, right? So that means for incomplete dominance, the basic here is that the dominant allele, unlike the complete dominant, does not have the full dominance over the recessive. As a result, instead of simply choosing red or white, you have a third option, as you can see on the picture here. Instead of having red for the heterozygous, it will actually result to the combination of the first two alleles. The combination of the dominant, which is red, and the combination of the recessive, which is white. So that means you have now three options: red, white, and pink. Sa dominant, di ba, remember sa complete dominance, your option were only red or white. Do you see the difference? Okay, later on, we'll have application on that. Now, the other one, which we will learn yet tomorrow, so yesterday, tomorrow, is about codominance. So I'll just give you a teaser on that. I will explain it later on, tomorrow. All right? So this one, codominance. Co-worker, co-founder, there is a sense of being together, being equal. So I'll explain that tomorrow. So let's now focus on incomplete dominance. So there is a video on our module. There's a video link wherein you can use that as your reference to watch what we mean by incomplete dominance. So, as I've mentioned a while ago, or and as I what I mentioned in our in my analogy, one important um unique characteristic of an incomplete dominance is that it will give you a third option, and it is actually the blended or the combination of the two alleles. Red and white will result to pink. Okay? So it does not resemble the pure dominant and the pure recessive. So let me give you an example here. Let us say blue allele or the blue option is the dominant, and then white is the recessive allele. Now, please take note of this one because in terms of the coding for incomplete dominance, there's a slight difference. Okay, there's a slight difference in terms of how you write the coding. For incomplete dominance, for the what we will do is get the first letter of the dominant allele or gene. In this case, the first one is blue, so we will have capital B. Kung complete dominance pa ni siya, we will simply use the same letter and make it a lowercase. But since this is incomplete dominance, the way we represent the recessive allele is this time the capital letter of its own description. So blue is dominant, white is recessive. But when we represent them in terms of their code, we will use their individual first letter. B for blue, W for white. Is that clear? Okay? So, dili na siya different in terms of the cases. They're actually different letters now. Nasabtan? Was that clear? Okay? I hope that's clear to everyone. Okay. So, that means if you see the letters B and W on the genotype, that means it does not resemble the pure dominant or homozygous dominant, and it will not resemble also the homozygous recessive or the pure recessive. Instead, it will result to a blended characteristic. So what's the color combination when you combine blue and white? Of course, it's either you describe it as sky blue or light blue. So here, capital B is a homozygous dominant. What you see on the plant, you have blue petals. WW, that's homozygous recessive. What you can see on the organism, it is all of the petals are white. But if the genotype now is a heterozygous, a combination of two letters, B and W, it is now the third option, which is neither blue or true blue and neither true white or pure white. It is a combination. So I hope it's clear on the screen, on your screen, the difference between this one. This is a bright blue and this one is a faded blue. Klaro ba siya? Can I have a thumbs up if you can distinguish the color? Okay. So this is darker compared to this one because of course this is now with white. Let's now do an exercise. Okay, this time we're talking about a plant called 4 o'clock plant, and it has actually a flower. And then red is your dominant and white is your recessive. And this particular plant actually observes or follows incomplete dominance. So how do we write a red flower 4 o'clock plant? What's the first letter for red?
R.
R. And since it's a homozygous dominant, sorry, it should be, sorry, ano siya mga gawas for a while, ha? So it should be, how do we write that? It should be R capital, R capital. Very good. Yes, Marinel. Now, how about for white? What's the first letter for white? W. So white will have a homozygous white. So W, W. Now, we have a third option. This time, the plant can also exist or the plant can also have a pink flower. The question now is how do you write the code or the genotype if you have this flower? Yes, how do we write?
Big R, small W, sir.
Very good. Correct. So it is actually the heterozygous. One capital R and one capital W. Very good, Ian. So homozygous dominant, homozygous recessive, and this is your heterozygous. It's still a state of being different, but this time the difference now it's not limited to the cases or capitalization of the letter, but they are actually different letters. Okay? I hope that's clear to everyone. So that's how we will represent them. That is only applicable for incomplete dominance. So teacher, how will we know that that particular problem is incomplete dominance? There is a statement or it is stated as part of the problem. So let's have here problem set number 1. By the way, the problem set, same thing. It's found on our module. So we have here another plant called a snapdragon flower. Red is dominant while white is recessive. And if you have a heterozygous, that will give you pink. So let me give you an example. Okay, so this is the actual image of a snapdragon plant showing the different colors of its flower. You have red as the dominant, white as the recessive, and the blended option is pink, meaning it's the um, heterozygous genotype of the plant. So let us answer the following question. So we will start with the genotype of the parents, and then we will do the cross and then answer the other 3 questions. So let's start first with the trait. The trait here is that it talks about the flower color of the snapdragon plant. So what are our options or alleles based on the problem?
Red and white.
Red and white. And take note, because this is incomplete dominance, pink is also another option. So your parent here, so if you look at this short statement, your parents, okay, the parent plant are both snapdragons with pink flowers, meaning they are both heterozygous. So what should be the genotype of parent 1 and parent 2? What should be their individual code?
R and W.
Okay, very good. So that means parent 1 is pink, RW. Parent 2 is also pink, RW. Now, it's very important that you understood this part because take note, this is our springboard or starting point for the next part, which is for the Punnett square. So let me give you this one. Okay, RW for parent 1, RW for parent 2. Now, what is the result on the first quadrant here?
RR.
Yes, RR. But what is the color of that flower? Red, white, or pink?
Red.
Very good. This will result to red because it is a homozygous dominant. How about on the second quadrant here? You have RW, RW. What is the flower if you have this genetic code? Pink, red, or white?
Pink.
Okay, pink. Very good. That's pink. This will result to pink. Now, what do you think is the genetic code for this Punnett square or this style? What's the genotype? The letter combination.
RW.
Okay, also RW. And what would be the color for that? Red, white, or pink?
Pink.
Pink, another pink. And lastly, on the final or fourth quadrant, what's your um, genotype? The genetic code. Yes,
W.
Ian?
W white.
Okay, very good. Kompleto na kay Ian. So, WW at the same time white. So, how many combinations do you see on the Punnett square? 1, 2, or 3 in terms of the letter combinations? How many
3.
combinations? 3.
3.
Very good. So, that means all of the options are actually found on the Punnett square. So, we have a homozygous red, heterozygous pink, and a homozygous white. So, of course, we simply write the individual genotypes. Red, pink, and for white. Now, the question here is, what is the probability or the chance that you will have pink? looking at the individual squares. Okay,
50.
correct, Sophia, Marinel, Rupert. Yes, correct. So that means looking at is, remember, RW is pink. That's a combination of red and white. And based on what you can see on the Punnett square, there are two RW combinations on the four squares, out of the four squares rather. So that means you have a 50% chance. So what if I ask you which color combination has the highest probability? Pink, white, or red?
Pink.
Pink. Very good. So 50%. How about lowest probability or chance?
Red and white.
Okay, red and white. So that means you already understood the concept. All right? So that's for problem set number 1. Any questions before we go to the second problem set? Any questions? Wala. Okay. Let's go to problem set number 2. This time, we're not talking about um, plants. This time, we're talking about a certain species of birds. So we have black feathers are incompletely dominant to white feathers. Now, your given parent here, or your trait here is about, of course, the feather color. And your options are black, white, and take note that you must also specify that it's incompletely dominant. Black is incompletely dominant over white. Okay. So let us see if we understood this here. You are given three parents or parent combination. So take note, we have black, we have white, and we have gray. So, first set of parents. Mother is gray, father is black. How do you write their individual genotype? So, that would be BY for gray, BB for black. Okay, that's your guide. Now, how about for the second set of parent, wherein both of them are gray or has gray feathers? How do you write their genotype?
BW.
Perfect. So, parent 1, BW, parent 2, BW. And lastly, what about if we say we have the same mother, gray, female, but this time we have a white father or father with a white feather? How do you,
BW.
WW.
and
WW.
KWW. So, more or less, again, this is the crucial part because once you don't get this correctly, the next step will be very difficult for you because, of course, these are the codes that we will use on the Punnett square. Okay, so let's start with the first set of parents. Okay, one is gray, one is black. So for the first quadrant, what would be the color or what would be the genotype? Okay, very good. And that will give you black. Hindi na ako isulat ha. Okay. How about this one? What's the color if you have BW?
Gray.
Gray. Or gray na may E or gray with A. Actually, it's the same. It's both acceptable spelling for gray. I think British lang ang E. A is American English. Anyways, how about this one? BB, that's black. BW, that's gray. Now, how many combinations do you see on the Punnett square? 1, 2, or 3?
2.
Okay, it's limited only to 2. BB, BB, sana all BB, and BW. Alright? So, walay white. walay white here in this particular set of parents. So we have a homozygous black and a heterozygous gray. So of course, BB, noon BB kanya. BW, ngayon hindi na. BW. Now, the question now is, what's the probability or chance that you will have black since nara may duha ka options sa atong Punnett square? How many or what's the chance? Okay, very good. Candy. It's actually they actually equal. 50% black and 50% gray. So if I ask, is there a chance that the offspring will have white feathers? What would be your answer?
No.
No, it's an impossible event, or wait, or it that you can also state it as an impossible event, or it's not possible to happen given this set of parents. Okay, question for the first set of parents. Clarifications? Wala na? Okay. So let's now go to the next set of parents. Let us see if this time naabatan nan options. Okay, we have gray mother and white father. Ay, sorry. Ah, sorry. So we have BW, BW. Ay, sorry. Third set naman eh. Sorry. Nawala siya. Okay, BW and white, white. So, looking at our given set of parents, what's the probability here? Or what's the combinations? We have B, W, 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 and another BW, and another BW, W. So here, how is this similar to the previous problem? Do you see the pattern here? Or do you notice anything similar to for uh, anything similar in comparison to the first set of parent?
Yes.
Pareha sila 50-50, sir.
Okay, very good. So here, on this given set of parents, you cannot actually expect to see black, white, and gray sa mga offspring. Duha lang out of the three possibility. So the question in this set of parents, it's is there not is blah, blah, blah. In this set of parents, do we have now the chance to have white offspring with white feathers?
Yes.
Yes.
Yes. Kaganina, wala sila. It's an impossible event. So it depends, of course, sa given set of parents. Now, how about the third set? Both parents are heterozygous. Gray. BB, BW, BW, and WW. Now, looking at the Punnett square, how many combinations do you see?
3.
Three. So, in this set of parents, we can now see all of the possible options. Black, heterozygous gray, and white. So, what's the chance that the offspring will be gray?
50%.
Okay, that's 2 out of 4. You can also say 2 out of 4. How about black?
25, 1/4.
Okay, 25 or 1/4. And how about white?
1/4 pod.
Okay, 1/4 kapon. Okay, so that means it really matters the given set of parents so that it will actually dictate or limit the possibilities or the possible combinations for the offspring. But of course, we're only talking about one characteristic here. And of course, all living organism, we have millions of characteristic in us. Sa mata pa lang daan, ang shape, ang kabagaon sa imong kilay, imong pilok and on so on, color sa imong mata and so on. So isa lang na ka portion sa imong face, daghan na kay siya trait that you can observe. How much more if you're talking about the entire being of a person. So it's really that means it's really impossible for us to have one two identical individuals. Yes, they may have identical traits, but not all traits. Like all of yun. Even gali ang atong mga fingerprints, they are also unique. So wala gyud identical. Unless of course if you are a clone. But of course, cloning is considered unethical if they are done among humans because dili siya allowed. Okay. It can only be done among plants or among animals. All right? So are there questions, clarifications? Okay. So if that's the case, I'll see you tomorrow. And this time we will be focusing on the non-Mendelian genetics, particularly codominance. All right? So take care everyone, stay safe and stay warm and dry. Bye. Take care and God bless. See you tomorrow.
Bye, sir.
Bye,
Happy.
Bye.
teacher. Thank
Bye.
you.